Hello everyone, and welcome to The Explainer. Today we're diving into a massive, completely invisible industry that quietly keeps the modern world healthy. We're going to explore the hidden chemistry behind every single drug, vaccine, and therapy on the market today. It's a world of immense scale and just staggering precision, and honestly, I am so excited to guide you through the whole shebang. Let's get started.
Think about it for a second. Right now, somewhere out there in a climate-controlled warehouse that you and I will probably never see, sits a small glass vial of white crystalline powder. It has no brand name, no flashy advertisement. It's literally just raw chemistry. But every medicine ever made, every tablet, every injection, every infusion, begins exactly like this, just as a molecule.
You see, without these unbranded, raw chemicals, the life-saving cancer drug your family member takes wouldn't exist. There's just no way. The diabetes injection saving millions of lives simply wouldn't work. Even the vaccines that stop global pandemics, they never would have made it out of the lab. Every single treatment has to be synthesized, purified, and tested long before it ever touches a patient.
Section 1. The invisible industry. Okay, let's dive into this unseen supply chain that makes it all happen.
So every approved drug out there, whether it's a generic everyday pill or an experimental therapy in a cutting-edge clinical trial, follows this fundamental evolution. We start with research chemicals, right at the laboratory bench. From there, we move through process development. We scale up to a highly regulated API, and finally, we formulate it into the actual drug product you pick up at your local pharmacy. Let's walk through how this pipeline builds from the ground up.
Drug discovery always starts small. Academic labs, biotech startups, contract research organizations, they all use research chemicals. These are the fundamental building blocks, the reagents, and the solvents used to construct new molecules. And I love this analogy. A research chemical is like a single Lego brick. Quality matters immediately. If you start with a contaminated reagent, you can throw off an entire experiment, right? You're basically wasting weeks of work and thousands of dollars. You are laying the absolute foundation of discovery here.
Now, once a molecule actually shows real promise, it transitions from just a research-grade chemical into what's called an API, or active pharmaceutical ingredient. Think of it this way. The API is the car's engine. It's the part that actually does the work and produces the therapeutic effect. Everything else in that tablet, the binders, the coatings, the preservatives, those are just excipients. They're the chassis and the seats, essential for delivering the engine safely, sure, but they aren't what actually makes the car move.
But here's the catch. Making an API isn't like mixing up a small batch of a research chemical. To be used in a human patient, it absolutely has to meet good manufacturing practice, or GMP standards. GMP is kind of like a recipe that a restaurant has to follow exactly the same way every single time. Every process is documented, the equipment is qualified, and raw materials are perfectly traceable. The goal? Absolute batch-to-batch consistency. A pharmaceutical manufacturer needs to know with total 100% confidence that the API is exactly what the label says before it ever goes into a human body.
Section 2. The GLP-1 peptide revolution. Now, what's really interesting about this slide is the incredible, precise nature of what we're talking about.
First off, what's a peptide? It's basically a short chain of amino acids, and it's incredibly precise in how it interacts with the body's receptors. And just look at this number, $130 billion to $250 billion. That is the projected market value for GLP-1 receptor agonists by the early 2030s. GLP-1s are a class of peptides originally developed for type 2 diabetes, but they have become an absolute global phenomenon for metabolic health and weight management. I mean, the scale of this peptide revolution is just awe-inspiring. Demand is arriving so fast that it's outpacing what most supply chains were even built to handle.
But actually manufacturing these peptides at scale, it is genuinely difficult. Unlike your typical small molecule drugs, peptides are built one amino acid at a time, in a highly precise sequence. It's literally like typing a sentence one letter at a time. If you get one wrong amino acid or miss one step, you create what's called a truncated impurity. And this impurity looks almost exactly like the target molecule, but biologically, it behaves very differently. It's like a counterfeit bill that passes almost every single test. Catching it requires the most sensitive purification technology on Earth.
Section 3. High-stakes specialty molecules. Let's shift gears to the true frontier of precision medicine.
These are therapies designed for specific genetic profiles. We're talking targeted oncology, cardiovascular diseases, and rare conditions. And the physical challenges here are extreme. On one side, we have highly potent active pharmaceutical ingredients, which you see a lot in oncology. These are like highly concentrated spices. A tiny amount goes a really long way. That means they require intense hazardous containment facilities just to handle the raw form safely. On the other side, we have rare disease research. Here, patient populations might be under 5,000 people worldwide, so you're dealing with tiny production volumes, super fragile single-source supply chains, and literally no room for a sourcing error. Every single batch absolutely counts.
Section 4. The purity problem. This is where things get really high stakes.
So, how does anyone actually know what is inside that tiny, nameless file we talked about earlier? Well, welcome to the ultimate life-or-death security checkpoint, analytical chemistry. Analytical chemists are looking to answer two very different but totally crucial questions. First is identity testing. Who are you? This confirms the substance is actually what it claims to be. Second is purity testing. How clean are you? This quantifies exactly how much of the sample is the target compound versus any impurities. It's exactly like checking someone's ID at the door and then taking a deep dive into their detailed health records. Both matter immensely. To find these answers, scientists rely on a heavy-hitting analytical arsenal. You've got HPLC, which separates a sample into parts. Think of it kind of like sorting a mixed bag of marbles by size on a vibrating tray, searching for that single clean peak. Then there's LC-MS and GC-MS, which combine that separation with mass spectrometry to weigh molecules with just extraordinary accuracy. And NMR, nuclear magnetic resonance, acts essentially like an MRI scan for molecules, revealing the actual atom-by-atom internal structure without even opening up the sample.
You know, in pharmaceutical testing, you'll often hear scientists say, I don't trust one method, I trust two methods agreeing. For a lot of applications, purity specifications sit right around 99.5%. Now, that difference between 98% and 99.5% might sound tiny, but it is the difference between a glass of water that is almost clean and one that is truly safe to drink. Missing that threshold, it means the entire batch has to be completely discarded. No exceptions.
Section 5. Trust in the supply chain. So the crucial point is zooming out to look at the macro level here. Navigating the global sourcing of these perfectly pure molecules is basically an incredibly complex regulatory maze. Materials cross borders and pass through multiple intermediaries every single day. Listen, pharmaceutical sourcing is never just about finding the cheapest price. It is entirely about absolute trust and flawless documentation. A certificate of analysis, that's like a nutrition label for the chemical. Batch consistency ensures you aren't ruining your experiments. An FDA registration is basically the passport and visa for that chemical to enter a legally formulated drug. Without this paperwork, the whole process just stops dead in its tracks. The cheapest option is rarely the most affordable one if it causes months of regulatory delays. Because let's be real, navigating all of this can consume as much time as the science itself. That's exactly why the industry relies on trusted partners working quietly in the background. Companies like Abios provide this critical infrastructure, doing everything from delivering research chemicals in GMP-compliant APIs to running the exact independent analytical purity tests we just discussed. They handle the complex sourcing and testing ecosystem so that chemists, biotechs, and researchers can actually focus on doing what they do best, the science that saves lives.
It's truly fascinating when you pull back the curtain, isn't it? Every medicine begins as a molecule most of us will never see, made by people we'll never meet, and tested to exact standards we never even knew existed. So I'll leave you with this to think about. Go look at the medicine in your cabinet today and ask yourself, how many invisible labs, precise tests, and nameless vials of powder did it actually take for that exact pill to make it safely into your hands? Thank you so much for joining me on this explainer, and keep asking those great questions.